A test cross is an experimental procedure to explore the genotype of a particular individual organism. But in case of back cross, it is used basically isolating of certain characteristics in a related group of species. Hello friends, welcome to Biology Concern. I'm Animesh, and in this video I'm going to discuss about test cross and back cross in a brief manner so you can understand the whole topic very easy. So let's jump into the topic. First thing, test cross is a way to explore the genotype of an organism. So it is basically to explore the genotype of a particular organism, whereas the back cross is the mating of hybrid organisms of one of its parents with an organism of genetically similar to the parents. So basically here test cross is back cross. So in test cross it is a way to explore the genetic component or genotype of a particular organism, whereas in case of back cross it is used for isolating certain characteristics in a related group of animals or plants, that means related group of species. So this is the basic difference between test cross and back cross. So what is done by test cross and what is in back cross? So if we consider a long pea plant, that is the genotype of this long pea plant is homozygous and the genotype is capital T and that crossed with a small t phenotype, that is the short pea plant. So this is the short pea plant. So whenever we cross capital T into with small t, so capital T generates a gamete that is capital T, and this phenotype, short phenotype, generates a gamete with small t. Whenever we crossed these two genotypes, we get capital T small t. That means we get a long pea plant that is long in nature. So basically it is long, so it is in heterozygous condition, means in this case if this is a gamete, if this is a chromosome pair, so in this chromosome pair one is capital T, so one is for capital T, so this allele for capital T, and another allele is for a small t. So if we isolate this particular genotype from this organism, so we can do a test cross. Okay, so basically whenever we crossed this particular F1 genotype, so this is P1 genotype, P1 parental 1, and this is F1 genotype. So whenever we crossed this F1 genotype with the double recessive parent, that means capital T small t, so capital T small t into small t, we get a genotype of this particular plant. So if we get a long pea plant, but it is a type of unknown genotype, right? So basically in case of phenotypical dominant, so this pea plant is phenotypical dominant. Okay, so phenotypically dominant, so this is phenotypically dominant, but we do not know of its genotype, so it is basically unknown genotype. So unknown genotype. So if we have to find out the genotype of this particular plant. So whenever we want to figure out the genotype of this particular plant, we basically perform a test cross. So in test cross, this phenotype of or this pea plant is crossed with double recessive parent. Okay, that is small t. So the genotype for this pea plant would be capital T or capital T small t, but we do not know the actual genotype of this pea plant. So whenever we crossed this double recessive parent, then we get some phenotypical characteristics or phenotypical expressions of this particular pea plant in the next generation or F2 generation. So if it is capital T small t in nature, so if we draw a Punnett square, so capital T small t along with small t. So in this case we get small t and small t, but here capital T small t. So 50% of the individual would appear in the small type. Okay, so 50% appear in small phenotype. So whenever we get a small phenotype in the F2 progeny, so in F2 progeny or in F2 progeny, whenever we get small phenotype, then we easily find out that so this pea plant is actually a heterozygous type. Okay, so basically where it is stated that early use of this test was an experimental mating. So this is an experimental mating to determine what alleles are present in the genotype, and a test cross can help to determine whether a dominant phenotype is homozygous or heterozygous. Okay, so if this is heterozygous, this should be capital T small t. If this is homozygous, this should be capital T capital T. Okay, and is an experimental cross of an individual organism of dominant phenotype of unknown genotype. So dominant phenotype of unknown genotype organism and organism with homozygous recessive genotype, that is small t in this case. Okay, so this is basically a test cross. But in case of back cross, whenever we applied this theory, so we applied capital T into small t. This is the tall phenotype and this is the short phenotype. So we get small t gamete and we get capital T gamete, and this is the P1, and in case of F1 we have got capital T small t. Okay, so this is the hybrid. So this plant is hybrid, right? So if we want to isolate certain characteristics from this particular plant, we have to backcross it, and whenever this capital T small t is crossed with any of the parents, that is the capital T or capital T small t into small t small t, so any of the parent, whenever it's crossed with any of the parent, so it basically would be capital T and it basically a small t. So whenever, so this is the number 1 procedure and this is the number 2 procedure. If we isolate certain characteristics, like we want to isolate the dominant phenotype of this plant, pea plant. Okay, so whenever we crossed capital T small t with capital T, we get Punnett square. So capital T small t, capital T, capital T small t, capital T small t. So we get 50% of the dominant, that is homozygous dominant, and 50% is heterozygote. Okay, so whenever we have to isolate this dominant phenotype or this dominant characteristics, we have to play with this back cross. Okay, and if we want to isolate the short phenotype, so if we want to isolate the short phenotype in this case of test cross, the 50% of the F2 progeny will be, so 50% of the F2 progeny will be homozygous recessive gene in its, that means it is short type. Okay, so that means this is the gamete, and if this is the chromosomes, so small t and small t there. So we can isolate the dominant phenotype as well as, so we can isolate the dominant phenotype as well as the short phenotype in this case of back cross. So basically in animal breeding, a back cross is also called a stop cross, and it is a traditional way or traditional process that prevents new recombinations. In the back cross, there is no new recombination occurs, so there is no variation would be produced. There is a chance that old characteristics are present in this type of plants, but not new characteristics appears in the next generations. So this is basically a back cross. So this is the traditional way, but in the test cross, we want to explore the genotype of this plant or animal, so we can go through the test cross. But if we isolate certain characteristics like dominant or recessive or short or any other characteristics, so we can go through the back cross. Okay, so I think this video is helpful for you to understand the topic better, and if you like that video, please hit the like button, don't forget to subscribe, and thanks for watching.